Okay, so good afternoon, everyone. Thank you very much for hanging on in there and joining this webinar. And I hope that the festive season will start for you shortly afterwards. So this is going to be a two-part webinar. We're going to start with Dr. Kevin Ashelford. He's the lead for strategy and IT infrastructure at the Wales Gene Park. And he's going to talk about how we can find ways together, really, to share data with and from the NHS. So Kevin's going to talk about this for sort of 10 minutes or so, and then there'll be an opportunity to ask him questions either via the chat function or by email afterwards. Kevin is happy to receive your questions by email after the webinar. Then Dr. Helen Pearson will talk about her research into targeting the Pi3 Kinase Pathway for treating prostate cancer. So Helen is currently at ESCRI, so that's in the School of Biosciences, and her research is supported by a career development fellowship from Cancer Research UK. So Helen is therefore a fantastic example of a very successful early career researcher and group leader here at Cardiff University. So Helen will talk for about 30 minutes and will also be happy to receive your questions, again receiving the chat function. So without further ado, we pass on to Kevin. So Kevin, do you want to share your screen with us?
Yes, let's give this a go. First hurdle to get over. And hopefully,
perfect.
Excellent. Rightio, so I'll just, whoops. So yeah, so basically this is a short 10-minute talk really just to give a brief overview of data integration and how we as cancer researchers can be working with data controllers like the NHS. And what I'm going to do in this talk really is several things. So I'm going to focus on the fact that we do need, what do we need to do to share and combine and exploit medical data as researchers. I want to look at the challenges that we face if we are going to do that in the longer term and also short term. I'm going to be introducing you, and this ties in with the short term aspect, the Circumary IT project, which some of you may be familiar with, and it's something that is currently being actioned. I will touch upon the challenge of information governance and security. I'll highlight the need for case studies for us to collectively be developing our strategy. And then just a very brief look to the future as to how are we going to enter our glorious data integration future. So let's start with that future. You know, when I was putting this together, I very briefly thought, what would be ideal would be just to demonstrate, put him on a screen where we want to head for, that final destination. That destination is going to be one where we're going to have all the compute that we require, we'll have the storage that we need, we'll have the software that allows us to utilize that infrastructure and apply it to the data that we may have. That would be in some sort of trusted research environment. That's the terminology that tends to be used these days. And we'd also want to access data from other data controllers elsewhere, for example, the NHS or industry and so on. And inevitably, we as researchers would need some way of accessing that. Now, that's the future, that's the vision, and that's easy enough to put down in a schematic. It's a lot more difficult to actually realize in detail. And the reason for that is that, well, there are a lot of things that are outside of our control. We need to be working with partners. We currently lack the resources we do need. We need to be working towards that point. And so with that lack of clarity, how do we plan for the future? That's really what I'm going to try and grapple with here. And grappling with that uncertainty, well, we can actually identify some certainties that we can cling on to. One of those is that we must work with multiple data controllers. In other words, other organizations or entities that own data. The NHS is an important example of that, but there are others as well. So we have to find ways of working with the NHS and others. We also must find ways of cooperating with other local, national, international organisations, both academic and commercial, in order to achieve our data integration aspirations. That means organisations like SAIL and UK SERP, DataCan, the UK-wide data hub that's been created by HDR UK, and so on. We also are clear what data we're interested in: genomic data, imaging data, free text data, routine clinical data. We know what these data types are. And as a consequence, we do know what the information security and governance issues are. And these are important that we need to be focusing on. And finally, we all know we have to invest in data science skills. So what I've listed here is a set of certainties that we can focus around. And in order to deliver on those certainties, one thing we do need to be doing now is focusing on the standards and frameworks we need to be interoperable. In other words, to be able to talk and communicate with the NHS and other entities. And we need to find how to work with those partners. So for example, SAIL, how to work with SAIL. NDR, the National Data Resource, which is the NHS's Wales' new sort of way of organising its data. Now it's a 10-year project that's been started, and they have mechanisms called APIs that allow one to access their resource. And we need to understand what they are. DataCan I mentioned earlier, which is part of HDR UK. And then there are international standards that we need to be grappling with. So for example, in the realm of genomics, GA4GH is a group of standards that we really need to be embracing. So what I'm listing here is just a few things that we certainly need to be focusing on now. I've talked about certainties. I think we can also make some reasonable assumptions of where we're heading. One of those is that the future trusted research environments that we'll be working on are unlikely to be local. We're probably going to be working with some third party, whether it's in Swansea or commercial provider or whomever. So that's something we need to bear in mind. Federation in some form will be the future. Now, federation is a term that's increasingly being used. All you need to be aware of here is that this image shows three sort of categories of data storage. The one on the far left in blue, that's a traditional way of organizing data. You put all the data together in a single resource, and that's what SAIL represents. Federated systems link together multiple data centers. And in the green section of this image, it shows a form of federation where databases link together. And a second form of federation is the one indicated by the red schematic. And that's where instead of moving the data to the analysis, you move the analysis to the data. I won't go into detail about federation, but it is something that we're going to have to be aware of and cope and deal with. The problem is it's not clear at the moment exactly what form that will take. And so that's where the ambiguities start to creep in. Now, given the uncertain future, how do we deal with it? You know, there are all sorts of possible scenarios that we could be working with, whether it's ARCA, SAIL, UK SERP, Microsoft, Google, Amazon, or so on. At this stage, we just simply don't know which is the best way to move forward. So we need to develop a strategy that is platform agnostic. It doesn't really matter where the compute and storage is. What matters is our ability to communicate with our partners and use the right standards that allow us to be flexible. And so that's really what we need to be focusing on in terms of a strategy. But I want to now focus, that's the picture, that's the future. But I think shorter term, there's more immediate requirements. We need now dedicated compute and storage. We need, moreover, it needs to be something that is secure, that has the right standards that NHS Wales and other bodies want to see in order to trust us with their data. We need something that can allow us to attract further funding. And we need something that we can start laying the foundations. And that something is in the form of the Succumry IT that I mentioned right at the beginning. Now, the Succumry IT is £300,000 worth of investment from Welsh government in cancer and rare genetic disease research to build compute infrastructure that we can then use. It's going to be built in the Redwood Data Center alongside Arca Hawk. It's going to consist of firewall and switches, login node, management node, compute nodes, and most importantly, perhaps storage. And that's replicated storage, so it'll be replicated in Park Place. So that's what we're actually in the process of building. We're procuring it at the moment. Just to give you just a little bit more background, Succumry IT is actually a collaboration between Wales Gene Park, focusing on genomic data, the Wales Cancer Research Centre, Wales Cancer Bank, and several other partners, including Cardiff Cat and Colorectal SPED. And together, we're trying to use this infrastructure to focus on machine learning. Now, machine learning is really just an example of the sort of data science we're interested in. Basically, we want an infrastructure that can allow us to analyze linked data, and that's what this is all about. I mentioned that Succumry IT is about laying foundations, and I just want to give a little example of what I mean by that. What's very important, I think, to a lot of us is data storage. And this table just illustrates the cost differences that we might face and how we need to plan for the future regarding those differences. So let's take, for example, consider you want 100 terabytes of storage over 5 years. Now you could do that by buying a heap load of external drives. It costs around £2,000. Now that is what you could do. That is a really, really, really bad idea. It's bad because you don't have any data resilience. It's bad because you don't have any data replication. It's really bad if you're handling medical data that's in any way sensitive. If you're doing that, then you're basically breaking universities' data governance regulations and possibly more than that. So we really don't want to do that. But I'm putting this figure up to show a comparison because the university alternative, university RDS alternative that's still being developed, it comes in at a whopping great 83 to 104K over a 5-year period. Now, and the reason it's expensive is because it provides the security and data resilience that we need. But it comes at a heck of a cost, and that is not sustainable, I would say. I don't think any grant being developed at the moment can afford to put that sort of money on it. So we have to find a way of getting that money down, that price down. As a comparison, Microsoft Azure, a Google, cloud provider, it comes in at kind of a similar price. It's difficult to get these figures. There's all sorts of hidden costs, but this is a rough guide, 90K for 100 terabytes over 5 years. Now, with the Succumry IT, we've actually managed to get the price down to around 30K for 100 terabytes. And I think we're trying to aim for is saying around 25K, because I think it's that sort of level is a realistic level for dealing with, for buying the sort of storage that we're all going to be needing. So I put this slide up, really just to show you how we do need to be making sensible IT choices now so that when we come to expand our storage, we're doing so at a level that is actually feasible, is sustainable. Now, I'm actually a little bit over time, so I'll just cover the last bit, which is case studies. I said that the Succumry IT is there to develop further projects, to attract further funding, to develop our resource and attract also more staff, the expertise we need. And so what I mean by case studies, well, here's a project that I've been working with the Wales Cancer Bank, and there you don't need to focus on the detail, but the point is that Wales Cancer Bank would like to sequence a whole slew of tumour samples, and the resultant data that's generated by the NHS wants then to link that with other data types, tissue pathology, image data, for example, other clinical data, and create a single database. And then from that database to create data sets that can then be fed into a machine learning algorithm. As I say, you don't need to focus on the detail here. The point is, there are various steps in this process that if we have specific case study examples for, we can start to flesh out the practical challenges of delivering that vision that I mentioned, that future that is still a little bit on the hazy side. So that's where I want to end it. I just want to end with your instant guides to some New Year's resolutions that I think we ought to try to embrace regarding data integration and IT. The first of those is we simply cannot work in isolation. Talk about cancer here, but requirements I've been talking about transcend disciplines, and we really do have to be finding ways of working much more broadly. Secondly, we as researchers have to take the initiative. We can't rely on university IT to solve our problems for this. We have to be driving them. Thirdly, we must be talking to each other a lot more. We need to be sharing our IT plans a lot more. We tend not to do that. We tend to work in our silos. We've really got to try to stop doing that. And lastly, we do need to think sustainably. We have to think, if we're storing data, for example, and we're storing it more than 5 years, that goes beyond warranty of a typical infrastructure. We need to find ways of storing longer term, and how do we do that? These are not our easy answers, but things that perhaps in the new year we can start thinking about. I'm woefully over my 10 minutes now. I shall stop. If there's any time for questions, more than happy to answer them. Thank you very much. And I'll also stop sharing.
Thank you very much, Kevin. So do we have any questions from our panel, our attendees? So I want to ask you, Kevin, about benchmarking. Are there other countries, organisations that are doing something similar to this that we can look at, you know, how they're doing it, and perhaps, you know, adopt certain sort of practices?
Oh, absolutely, yes. I mean, we don't want to reinvent the wheel, absolutely. And there are lots of examples we can be following. One example I can draw on straight away in the genomics field is, of course, Genomics England across the border. Very important. We need to be learning from them. The GA4, GH standards that I was alluding to are standards that actually the Genomics England are applying. There are other organisations across the globe who are all applying these standards. And you can look elsewhere in imaging, for example. Emiliano Spizzi, who is part of this bid, this secondary bid, Cardiff CAT, he is working on sort of standards, applying standards for federated machine learning of image data. So there's a lot that we can learn from, and I think it's a case of, you know, identifying those and following them. But yeah, absolutely.
Brilliant. Richard Adams is asking, what staff skills do we need to get this going? Are they in the university already, or do we need new skills brought into play?
They are in the university already, but they are very dispersed and not easy to get hold of. The skill sets we're talking about are fairly standard IT skill sets. But so, for example, the sort of data standards I was talking about is all about applying APIs, which is something that data scientists are very familiar with. It's all about applying things like Python and R and other scripting languages to perform the analysis. So the skill sets are around, but we do lack focus points for them. And I think initially we do need to be focusing on those data standards, how to implement them, those frameworks, how to implement. So we need skill sets that can implement those. I see kind of three broad categories of skill set for the data scientists. You've got the analysts, who their R and Python and other programming languages are really valuable. Then you've got the data database IT people, and there's a particular set of skill sets there. And then there's those that needed to glue the thing together, the standards application, that sort of thing. So yeah, there's a lot of skill sets we need, but it is present. But we do need a bit of
consolidation of these skill sets.
Yeah, and we do need more staff to sort of have those skill sets that we can access.
Yeah. And by having the case studies, then this brings to your attention which skills you need to solve a given problem, right? So is your talk, can it be seen as an invitation now to our attendees to contact you regarding some case studies of their own, right?
Absolutely, yes. It's anything that you, any sort of data, big data requirement that you have that could potentially be a focus for this activity. It's with the potential perhaps of working up into a full grant application if that's appropriate, or if it's a smaller piece of work. The Wales Cancer Research Centre are investing in some data scientist posts in the coming year, and those posts will, you know, they can be deployed to a certain extent in these sort of areas. What we need is a focus, and what the talk has tried to do is show you where that focus is. But anybody who has any case studies, any big data requirement, I'll be very interested in hearing from, because those case studies will highlight the practical, real requirements we will need to address. Absolutely. So there, yeah.
How to oil the wheel, so to speak.
Exactly.
John Chester has written on the chat, that was a super talk, Kevin, and I second that. This is a fantastic opportunity. So I hope the attendees take that on and also go and tell, you know, their colleagues and friends who weren't able to make the webinar, because they will actually be able to access this as a recording later anyway. Fantastic. Hey, thank you. Thanks a lot. So Helen, can I ask you now to share your screen, please?
Yeah, sorry, just loading it now.
So we have to be particularly grateful to Helen for agreeing to speak today, because she, in fact, it's not just the run up to Christmas for Helen, but she's also just moved house. So well done on all the multitasking, Helen, and thank you very much for doing this today.
No problem, thank you. Just lucky you can't see all the boxes around me. Well, first of all, yes, thank you for everyone sharing your afternoon, especially in the run-up for Christmas, to hear my talk about targeting the Pi3 Kinase Cascade to treat prostate cancer. I put up this slide as part of my title page because it's actually AKT staining in prostate cancer, and this is sort of all where it all began. Every single advanced stage prostate cancer will be expressing lots of phosphorylated AKT, indicating that the Pi3 kinase pathway is activated. And this therefore presents such an interesting therapeutic targeting approach for prostate cancer, as we could potentially capture nearly all advanced prostate cancer patients by taking this approach. So, just need to move my slide, there we go. So I know I had quite a broad audience today, so I've just put in an overview of what the prostate actually is to begin with. So the prostate, it's a gland of the male reproductive system. It sits just at the base of the bladder here, at the neck of the bladder, and it's about the same size as a walnut, so it's quite a small gland. It's growth stimulated by androgens, which is a male hormone, such as testosterone or DHT, dihydrotestosterone. And it functions to generate about 30% of the seminal fluid, and it provides sperm transport and nutrition. And the figure here on the right is a cross-section through one of the prostate ducts. So you can see the main principal cell type is the luminal cells, shown here in like a light pink. And they're supported by basal cells, which form a continuous layer, which are then surrounded by the basal lamina. And there's lots of different markers that we're using to label these different types of cells. And these luminal cells will then secrete components of the seminal fluid into the lumen of the duct. So prostate cancer, current data predicts that 1 in 6 men will be diagnosed with prostate cancer in their lifetime, which is gradually increasing. I think when I started my PhD back in 2004, it was actually 1 in 9 men. So things are becoming increasingly more prevalent in terms of diagnostics, mainly owing to the fact that our capabilities of diagnosing men with prostate cancer have improved, and the awareness of prostate cancer in men has also improved. So sadly though, it's still the second most common cause of cancer mortality in men globally, accounting for over 350,000 men male cancer-related deaths worldwide. And this chart just represents a treatment landscape overview from the past decade. So if you came into the clinic with prostate cancer, you're diagnosed with localized prostate cancer, then you would typically undergo active surveillance or local therapy such as surgery or radiotherapy. Most men will do very well on that, given that it's quite a later life disease. Most people will outlive their cancers. However, around a third of patients within about 5 years will go on to relapse. And it's at this point that they will be given androgen deprivation therapy, or a type of hormone therapy where we withdraw all the androgens. And we usually do this using first generation type drugs such as bicalutamide. And these initially have excellent responses. Nearly all men will respond to androgen deprivation therapy, but it's an unfortunate scenario is that most of the prostate cells will develop resistance to androgen deprivation therapy within around 1 to 10 years. And while we've had some immunotherapies come into play, typically you'd receive androgen deprivation therapy by second generation drugs such as enzalutamide or abiraterone, which target androgen signaling or androgen receptor pathway directly. But once resistance has developed, really the next step would be chemotherapy. Typically in prostate cancer, that would be docetaxel or cabazitaxel. But generally we see resistance occurring within around 12 to 24 months. And this is known as chemotherapy refractory disease stage. So the emergence of castration resistance here is one of the key areas that my lab works on. But it's really the failure of the current standard of care, which is leading to the deaths in treating these late stage diseases, that we need to identify new ways to treat this disease. So some of the new advances in molecular imaging and genomics have really started to make huge strides in this area. And just focusing here on the genomic sequencing, multiple large-scale genomic sequencing of prostate cancer patients has been able to identify actionable targets. And this is just a figure from a Nature Genetics paper from one of the larger prostate cancer databases that came out in 2018. And you can see here that some of the most commonly deregulated pathways in prostate cancer are the Pi3 Kinase Pathway, DNA repair pathway, also epigenetic regulators, cell cycle, Wnt signaling, and RAS signaling. So my talk today is going to focus on the Pi3 Kinase Pathway, but we do have projects looking at DNA repair, the Wnt pathway. We've just had a project funded by the Prostate Cancer Research Group, and also we do have work looking at RAS-mark kinase signaling as well. And I guess the other thing to mention on this slide is that we see much more deregulation of these pathways in the metastatic population of patients shown here in red compared to just the primary tumour in blue. So it's an evolving treatment landscape at the moment. Some of the key areas that have improved dramatically are how we treat metastatic hormone sensitive prostate cancer, so patients that present in the clinic with metastatic disease that haven't had any hormone therapy. There's now new systems in place to treat them with androgen deprivation therapy, with radiotherapy, or other androgen receptor pathway inhibitors. We've also, given the range of genetic studies that have come into play, we've also been able to approve new biomarker-driven clinical studies, such as the PARP inhibitor studies, for homologous recombination repair deficient patients. So those would be patients that are carrying things like a BRCA2 or BRCA1 mutation. So PARP inhibitors have been formally approved. We've also got a number of clinical trials looking at targeting AKT inhibitors, pembrolizumab as well for mismatch repair or microsatellite instability carrying patients, or there's also a new range of androgen receptor pathway inhibitors, such as the androgen receptor PROTACs. We've also seen quite a lot of development in the area of lutetium PSMA, so radionucleotide targeting using PSMA to target prostate cancer cells. So I think together these are showing promise in the clinic, and they are able to improve survival rates. However, there's still huge strides to be made because we're not really improving survival rates that much at the moment. So some of the key challenges for prostate cancer research that we need to address if we're going to improve our management of prostate cancer patients and benefit them in terms of survival, will be to develop a better range of prostate cancer models that recapitulates the full spectrum of the disease. It's not just one disease, there's lots of different subtypes, and we need models that represent all of these subtypes so that we can find better ways to treat each of those different subtypes. We also need to develop a better understanding of the molecular mechanisms underpinning prostate cancer. And with that, I guess we also need to understand how resistance occurs in the clinic to the current standard of care that we have, and also to identify better predictive biomarkers for prostate cancer so that we can identify treatment responders and treatment non-responders, for example. My lab really focuses on these in several different ways. So in terms of developing new models, we are specifically keen on developing new in vivo models that recapitulate the range of different disease subtypes by generating new genetically engineered mouse models, so transgenic mouse models, but also patient-derived xenografts. And we've also been establishing ex vivo organoids and explant cultures so that we can model the disease through more high throughput screening type-based approaches. In terms of the molecular mechanisms, we're very focused on understanding how Pi3 kinase signaling, DNA damage repair, and the Wnt signaling pathway, the canonical and the non-canonical Wnt pathways, contribute to prostate cancer disease initiation and tumour progression. But we're also interested in the therapeutic resistance arms through the Pi3 kinase directed therapies, PARP inhibitors, how we can improve the efficacy of targeting Wnt signaling. We're quite interested in understanding how therapeutic resistance stems from tumour microenvironment as well, and also linking all of this back to androgen deprivation therapy, as this is one of the most standard of cares for advanced disease. In terms of predictive biomarkers, we're keen on understanding whether the Pi3 kinase genetic drivers that have been identified in prostate cancer have predictive value, and also whether there's components within the tumour microenvironment that could also be predictive of tumour response. That's kind of an overview of what we do. I'm not going to be able to talk about all of that today, so I'm mainly focusing on Pi3 kinase with a few little snippets here and there on some of the other stuff. These are the main experimental approaches that my lab uses. So we've generated lots of different genetic engineered mouse models, including those of Pi3 kinase, which I will be talking about today, but also things like LKB1 loss, which is polarity regulator, and also beta-catenin, which is the master component of the Wnt signaling pathway. Basically what we can do with these gems is we can start to take out the prostate tumours and dissociate them and grow them in matrigel to form tumoroids, or we can slice them up and form explant cultures, which enables us to do lots of different preclinical studies. Or we can take patient samples and do the same, or we can take patient samples and put them into PDXs. But also through connection with the Wales Cancer Bank, we've also got the opportunity to look at biomarkers. Haven't done much of the blood-based biomarker work yet, but we have some ideas on what we might want to do with that shortly. But mostly we've been looking at tissue microarrays with a collaborator in Germany who has over 3,000 different prostate cancer samples that on a tissue microarray that we can pump through quite quickly. So the idea here is there to identify new biomarkers and correlate that back to the clinical follow-up data. And with all these models, we do a lot of molecular characterization, including things like RNA-seq, RPPA. We're moving into phosphoproteomics and metabolomics as well through the Rosetta platform based at the Pearson Institute up in Glasgow. And the idea there is to combine all of that information to identify new therapeutic targets and approaches, which we can then study using preclinical trials in these mouse model based or ex vivo model platforms that we've developed. And then obviously the idea there is to inform future early phase clinical trials. So this is the Pi3 kinase pathway. So Pi3 kinase exists as a heterodimer, and there's a regulatory pH5 subunit or a catalytic P110 alpha or beta subunit. So there's a couple of different isoforms here. So upon upstream activation by receptor tyrosine kinases, small GTPases, or in the case of the beta isoform, also G protein coupled receptors, the Pi3 kinase heterodimer is recruited to the membrane where it converts the phospholipid PIP2 into PIP3. And this then triggers downstream activation of AKT via PDK1, which phosphorylates AKT at phospho-308. And that then triggers a downstream cascade which mediates growth, proliferation, and survival. There's also mTORC2, which can be triggered by Pi3 kinase activation, which can hyperactivate AKT by phosphorylating at a second site, phospho-473. And P10 acts as a handbrake for the whole system, and it basically dephosphorylates PIP3 back into PIP2 to switch the AKT cascade off. I should say one of the major downstream effectors of AKT is the mTORC1 signaling pathway, which lots of companies have developed drugs to, which I will come to in a minute as well. So I think what's so important about the Pi3 kinase pathway is that it is so frequently deregulated in prostate cancer. There's lots of data out there from several different genomic and protein-based studies now suggesting that the Pi3 kinase pathway is deregulated in nearly all advanced cases. This is just an example from the larger Nature Genetics Memorial Sloan Kettering Cancer Center Dana-Farber data set, which shows that when we trawled for a panel of about 80 genes involved in the Pi3 kinase AKT and mTOR pathway, that 50% of those genes are altered in primary prostate cancer cases, and 75% of them are altered in metastatic cases, showing that this is a highly deregulated pathway. And obviously what's quite clear from this is that P10 loss is one of the most commonly lost genes in prostate cancer, and particularly so in the metastatic case. But then you've got all these other genes, and because P10 is so popular, most of the research is focused on the role of P10, but what about all these other ones? And that's really where some of the work that I'm going to show you about today is involved in sort of addressing what do these other genes do, and what's the importance of them in terms of their therapeutic responses. So this is an overview of the clinical trials looking at targeting the Pi3 kinase pathway in prostate cancer. So pharmaceutical companies have spent considerable effort developing a range of drugs that will target this pathway, given its involvement in lots of different malignancies, but particularly prostate cancer, where there's high prevalence of activation. So they've developed drugs that will target pan-Pi3 kinase beta specifically, the isoform beta, AKT inhibitors, mTOR inhibitors, or combination therapies involving both. But I think what the take-home message from this slide really is that given that there's been around about 40 different clinical trials, we know that around 76% of those have been proven to be ineffective and or toxic, and that's really due to the inability of the drugs to completely switch off the pathway, owing to redundancy within the pathway and forward feedback loops which keep it switched on. And there's also a handful of trials that are still active that haven't returned results yet. And some of them are starting to show promise, and some of them are also starting to show signs of not being effective. So I think at the moment, probably the one to watch is ipatasertib and capivasertib, the AZD5363 from AstraZeneca, which are the AKT inhibitors, mainly showing promise in the P10 deficient population. So we didn't really used to focus on which patients were going to be treated with which type of drug, but more recently I guess, stratifying patients based on their P10 status is proving to be more efficacious in the clinic. So more clinical trials are starting to stratify based on P10 deficiency or not, which I think may actually improve the outcome for targeting the Pi3 kinase pathway. However, like I said before, there's more than just P10 involved. So I think it would be important for us to start considering all of the different types of genetic alterations that are known to drive activation of the Pi3 kinase pathway and how they're going to respond to these types of drugs as well. So yes, as I mentioned before, P10 is one of the most common ones, but what about all of these other ones? So most of the work in P10 has been shown that P10 gives rise to invasive prostate cancer in the mice. It's been the main focus of research. This is a normal mouse prostate epithelium. If you lose P10 specifically in the mouse using a transgenic approach, then you will develop a hyperplastic lesion within about 4 weeks, which gives rise to prostate intraepithelial hyperplasia, which will then develop as carcinoma in situ and invasive carcinoma within around 3 or 4 months. So it's quite a quick model. Some people have seen metastasis, but it's not a reproducible finding. So we know it's locally invasive, but it doesn't progress to metastatic disease as you would see in advanced cases in the clinic. But what we became interested in really is, how do these different genetic drivers of the Pi3 kinase pathway mediate distinct and mediate distinct and targetable signaling events? So if we start to explore some of these different genetic events, we might actually be able to identify new ways and new targetable approaches for patients with these other genetic alterations in the Pi3 kinase pathway. But other questions that we had related to this are really about whether these different genetic alterations phenocopy one another. Do they co-occur? And if they do, does that have an impact on disease progression? Also, do they serve as predictive biomarkers? And what about all the non-genetic drivers of this pathway? So that there's lots of things that remain unanswered, and we're starting to dig a little bit deeper with some projects in the lab. So focusing on Pi3 kinase, this all really started in Melbourne, Australia, at the Peter MacCallum Cancer Centre when I was a postdoc over there. I became really interested in understanding what all these different genetic drivers do. And luckily for me, Wayne Phillips worked on the floor above, and he was a Pi3 kinase expert who just made a brand new mouse model, a transgenic mouse that was able to express a PIK3CA mutation, which is a known hotspot in mammary and ovarian cancers. So this is a really useful tool for our research. So we wrote some grants together to address the role of the functional importance of Pi3 kinase oncogenic mutations in prostate cancer. Basically what this mouse does is it expresses 2 versions of exon 20. One carries a H1047R mutation, where the other wild type one here just contains a stop cassette. And this wild type one is flanked by some LOXP sites. So when we use Cre recombinase to cut one of these LOXP sites, this whole segment is chopped out and the mutant one can then slide into place. And importantly, in the latent setting, this stop cassette prevents the expression of the mutant exon. So it's sort of a silent mutation in the latent setting. So the aims really were to determine if genetic drivers of the Pi3 kinase pathway that are present in prostate cancer but haven't been investigated previously can also contribute to prostate cancer initiation and progression, but also on top of that to develop new clinically relevant model of prostate cancer, because nobody's looked at these things before. We need to model them and see how we can treat them. So to gain a better understanding of the status mutations in prostate cancer, we analysed 9 different data sets that were available in CBio portal. Some were primary, shown in grey up here, and some were metastatic. And what the overall outcome of that was that PIK3CA mutations occur in up to 4% of prostate cancer patients, while copy number gain or high-level amplification can occur in as many as 62% of cases. So it seems that it's not that prevalent in terms of mutations, but 4% is still, quite a large proportion of patients that are undergoing treatment. And also with the copy number gain and amplification, which has been shown in the past to lead to increased activity of Pi3 kinase, is also highly prevalent. So this was just analysing the mutational status, and you can see that mutations occurred in common hotspots that were also common to other malignancies like breast and ovarian. And you can see that the H1047R mutation, which was the mouse that Wayne had developed, is also one of the key hotspots in prostate cancer. And when we looked at PIK3CA mutations, we found that it correlated to several different disease characteristics, such as lymph node metastasis and high-grade Gleason scores, but also found that it correlates with worse PSA recurrence-free survival. So it looks like this is a predictive biomarker for poor outcome in prostate cancer. So we took an approach to model this in mice. We crossed the Pearson mouse with the latent heterozygous H1047R PIK3CA mutant mouse. And what we found when we looked at aged cohorts of these mutant mice and compared them back to the wild-type Pearson only mice was that while the Pearson only mice have these nice uniform ductal structures, you see high-grade invasive carcinoma with 100% incidence in the PIK3CA mutant mice. And this was really surprising and amazing at the same time, because as far as we know, this is the first heterozygous mutation in a genetic mouse model like this to give this type of lesion. So we're pretty impressed with the fact that it had such a strong capability of driving invasive carcinoma. But I guess then came the second question really is, that if it can drive this type of tumour, how does it compare to PTEN loss? Are they the same, or are these two different genetic drivers of the Pi3 kinase pathway operating by different signalling mechanisms? So we used mice on the same genetic background and compared the PIK3CA mutant mice with the PTEN deficient mice. And what we found was that while both can give rise to invasive carcinoma, they have quite different phenotypes. So in the PIK3CA mutant mouse, for example, onset is much later than the PTEN model. They're quite normal for a longer amount of time. Tumour burden is a lot lower in the PIK3CA mouse, so I think it's about 2 to 3-fold the size of a normal prostate, whereas PTEN can get up to 13 to 15-fold larger prostate size. And the rate of progression is much slower in the PIK3CA mutant mouse relative to the PTEN deficient mouse. And obviously PTEN is still expressed in the PIK3CA mouse, but it's not expressed in the PTEN mouse. And we did a lot of work, which is all covered in the Cancer Discovery paper, which indicates that the PIK3CA mutant actually shows a high level of mTORC1 comparable to PTEN, yet the mTORC2 activity is very different. So if you remember, mTORC2 is responsible for hyperactivating AKT, suggesting that PTEN loss is able to hyperactivate AKT signaling to advance prostate cancer progression. One of the other questions that we became interested in is, can we target this using Pi3 kinase isoform selective inhibitors like the alpha and beta inhibitors? Did some experimental work to understand whether these tumours are driven by a different isoform. We took invasive carcinoma, we took mice with PTEN deficiency or a PIK3CA mutation when they developed invasive carcinoma, so sort of stage matched tumours. And we treated them for 4 weeks with either the alpha inhibitor A66, the beta inhibitor TGX221, or the pan Pi3 kinase inhibitor BKM120. And what we found was that in the PIK3CA mutant, the alpha inhibitor was able to reduce tumour burden, that's shown here as A66, which was comparable to targeting all isoforms with the BKM120, but the beta inhibitor didn't. So this suggests that the PIK3CA mutant mouse is alpha isoform dependent, which is kind of what we expected given that we'd mutated the alpha isoform. And then in the PTEN mutant, this was sort of an unexpected finding to some extent. in that the alpha inhibitor and the beta inhibitor alone did nothing, but when you combine inhibition of all of the different isoform inhibitors, you could reduce tumour burden using the BKM120. And that was also mirrored when we combined the alpha and the beta inhibitor. So this suggests that the PTEN deficient prostate cancer is codependent on P110 alpha and P110 beta isoforms. I guess this has therapeutic implications in itself, because it would mean that potentially an alpha or a beta isoform selective inhibitor probably isn't going to work in PTEN deficient cancer. However, I know that is actually something that is being done in the clinic for other diseases, but we would probably predict that that's not going to be successful, given the dependency on the other isoform. And I guess something else that became quite interesting for us was when we trawled through the genomic databases, 39% of the PIK3CA mutant prostate cancer patients also displayed PTEN loss. So we wanted to generate a new model to represent this cohort of patients, and also then to determine if these two different genetic drivers do cooperate by different signaling mechanisms. So if they did, then they might show synergistic relationship when we combine them in the mice. So we did this, and you can see even at 56 days, we saw a rapid regression to invasive carcinoma, shown here in green, in the double mutants of PIK3CA PTEN mutants relative to the single mutants PTEN, which only had hyperplasia at this time point, or the PIK3CA mutant, which was a normal, just like the wild type. And this advanced disease progression at this time point was associated with elevated proliferation and increased mTORC1 and mTORC2 activity. And we did some further work looking at androgen deprivation therapy here. And this is just summarized here, which shows that PIK3CA activated or PTEN deleted prostate cancer, if you castrate those mice, which represents androgen deprivation therapy and removes all androgen hormones, acquires castration-resistant prostate cancer over time. So by that, I mean that the tumours will initially shrink, but then they'll start to grow again, just like we've seen in the clinic. And that was associated with a further increase in AKT signaling. There's now a lot of work combining Pi3 Kinase Pathway directed therapies with androgen deprivation therapies to show, I guess, to target this raise in AKT signaling here. And there's some mechanistic information for how those two pathways crosstalk as well, but I won't have time to go into it today. And then in the combined PIK3CA mutant PTEN deficient mouse model, we see a sustained level of high level of AKT signaling and further inactivation of an mTORC2 downstream target. And these mice, importantly, when you castrate them, don't show any change in their tumour burden. So this is known as de novo castration-resistant prostate cancer, in that you don't see any response to the withdrawal of androgens. So that suggests that there's something about combining a PIK3CA mutation with a PTEN deletion that enables a tumour cell to evade any form of sensitivity to the withdrawal of androgens. And that's really exciting for us. And we've got lots of different projects looking at what that particular mechanism is, because that might be incredibly important to target in the clinic. So yes, to do that, we've been taking an RNA-seq approach, and this forms partly Manisha's PhD project. And she's done some initial analysis on the RNA-seq data and found that when you compare a PIK3CA mutant tumour with a PTEN deficient tumour, we see a huge NF-kappa B signaling response in the PTEN tumours relative to the PIK3CA mutant tumours. So this is gene set enrichment analysis, and you can see of the 20 pathways, about 60% of the pathways that were identified all correlate to NF-kappa B signaling. And when she pulls out the NF-kappa B signaling signature, we can see that there's some clear targets that we're quite interested in, like TOLAC2 and BCL3, which Richard Clarkson here has developed an inhibitor to. So she's busy exploring all of those and whether we can target PTEN deficient prostate cancer by targeting the NF-kappa B signaling pathway in combination with an AKT inhibitor. So the slide's not moving. There we go. So one way that she's doing this is the ex vivo explant culture. So to rapidly test for efficacy of the novel therapeutic approaches that she's identified. So we take tumours from GEMs or PDXs, slice them up so that they get embedded in agros and sliced on a vibrotome, and then they get popped into a culture dish on top of a little wedge of gelatin sponge. And this is an image of an H&E. So the beauty of this is that we're able to see the tumour microenvironment and the epithelial tumour responses all within one system for about, so far, we know 48-hour culture is viable. We're looking if we can take it further to 96 hours. But I think this is a really useful approach for some short-term assays to see if your drugs are going to have an efficacious response. And it enables you to have a look at the tumour microenvironment interaction at the same time, which I think is really powerful. Another model that we're using for this is also our newly developed neuroendocrine metastatic castration-resistant prostate cancer PDX model. And this was developed through our prostate MDRG pipeline in combination with the PDX platform established by Richard Clarkson and through the Wales Cancer Bank. So this is a real huge effort for everyone, and amazingly our first try at this has been successful. So what we had access to was a patient that came in for a right iliac lymph node metastatic needle biopsy, with a very small needle biopsy of 2 by 20 millimeters. And this biopsy was then segmented and implanted into 2 mice. So we had 4 segments into here, and we took a couple for next-gen sequencing and things like that, and RNA and DNA samples, and there was an H&E sample taken as well. What we found was that 3 of the tumours of the 4 that we implanted grew nicely, and they correlate nicely in terms of their histology as well. So this is the original patient biopsy, and this is the tumour fragment taken from, I think it was T3 in this case. So we know that these tumours correlate really well. And we've been using these in explants as well, and they're growing really well. And this is actually some data from another PhD student in the lab who's looking at DNA damage repair, and he's shown that at 48 hours, cleaved caspase-3 expression in the vehicle is quite low, as you'd expect. But when he treats with olaparib, the PARP inhibitor, he increases cleaved caspase-3. So we can use this type of system to do some very rapid drug screening and analyze the tumour microenvironment at the same time. So just lastly, and I know I'm right on time, but I'll skim through this very quickly. And it's Christmas is awaiting slides, which is about overcoming resistance to Pi3 kinase directed therapies. So a lot of the therapies that are in the clinic at the moment have focused on one arm of the pathway, of the Pi3 kinase pathway, and not the other arm. So this is something that I've been quite interested in for some time. So they either target Pi3 kinase, AKT, or mTORC1, but they haven't really targeted mTORC2. So I think this is one of the areas that our lab and others have seen, in that if you treat with a Pi3 kinase inhibitor such as the BKM120, you'll see some minor tumour regression, but you don't actually see a change in proliferation, and you certainly don't see a change in any of the mTORC2 downstream targets, phospho-AKT, serine 473, or in the SGK1 downstream target NGRG1. So, and that's all quantitated here. But I think overall that would suggest that the Pi3 kinase inhibitor resistant population is retaining NGRG1, an active mTORC2, SGK1, NGRG1 signaling in P10 deleted prostate cancer. And that's something that we've been following up in vivo and ex vivo models. And we're particularly looking at targeting AKT and SGK1 at the moment. And we've got some models to look at the functional consequence of deleting SGK1 as well. And then the idea there would be to undertake the molecular characterization, including metabolimics, to help identify new biomarkers and therapeutic targets and approaches. And just lastly, this won't take very long, it's to evaluate the role of Pi3 kinase signaling in the tumour microenvironment. We've also observed that as you delete P10 in the epithelium, you're also deleting P10 in the stroma. So this is a Pi3 mutant, where P10 is expressed in the epithelium, but it's also expressed in the stroma. And this is a P10 deletion mutant, where you can still see some brown staining of P10 expressed in the stroma, but it's not present in the epithelium. But when you when you activate Pi3 and delete P10 together, there's really hardly any P10 in the stroma. So that's become quite an interesting observation for us that we're starting to follow up to try to understand what the role of losing P10 in the stroma might be. And to start with, we're addressing whether stromal P10 deletion is a predictive biomarker for prostate cancer in the clinic. We're doing multiplex immunohistochemistry analysis with the Marafiotti lab at UCL. We've also got a tissue microarray analysis with Ronald Simon. His cohort is now up to 17,000 with clinical follow-up data, so it's an amazing resource, but it's quite an intensive, laborious task to sustain all of them, so we probably won't do all of them. But I think it'll be really interesting. Lots of people have looked at P10 in terms of its status in the epithelium, but no one's looked at it in the stromal populations. So that's something that we're going to start looking into. And also, when we delete P10 in the stroma using a stromal fibroblast specific CRE, what we see, which is really interesting, is that we start to see disease onset. And this is 56 days post tamoxifen induction, so 56 days since we induced the loss of P10 in the stroma. And you can see these hyperplastic disease lesions occurring in these mice, and importantly, they still retain P10. So I think there's something very interesting going on here. We've now got a platform where we can start to test what the role of P10 is in the stromal fibroblast population as well, which I think will be really interesting, because there's obviously some tumor stroma crosstalk interactions that are going on, which will be important for therapeutic implications for Pi3 kinase directed therapies as well. So that, I think it's sort of early days yet, but I do think that there's some interesting data and stories to be found out of this one that could inform clinical decisions. So overall, Pi3 kinase mutational amplification can predict for poor prostate cancer patient outcomes. Pi3 kinase HR activation is sufficient to cause locally invasive prostate cancer in vivo, and we know that that can cooperate with P10 loss to accelerate prostate cancer progression and CRPC growth. So potentially, if you are a patient with a Pi3 mutation and you've lost P10, then you're probably not going to respond to hormone therapies in the way that we would expect you to do so. And therefore, you're probably at high risk of developing a lethal form of prostate cancer quite quickly. And for you, those sorts of patients, we'll be looking at whether we can find a Pi3 kinase directed therapeutic approach to improve their survival. We also know that genetic drivers of the Pi3 kinase cascade aren't functionally redundant, but instead they drive prostate cancer via different signaling events. And each of those different signaling events can present a novel therapeutic target. And at the moment, we're very interested in understanding NF-kappa B signaling in a P10 deficient prostate cancer. And also that Pi3 kinase pathway multipoint blockade might prove to enhance therapeutic efficacy compared to monotherapies, and that the mTORC2 SGK1 signaling axis could present a novel targetable approach to overcome resistance to Pi3 kinase directed therapies. So determining how the Pi3 kinase cascade can contribute to prostate cancer in the context of all of these different Pi3 kinase genetic drivers that we've identified could identify new novel therapeutic approaches and aid patient stratification. And, you know, we are very interested in what the role of the Pi3 kinase signaling pathway is in the stroma as well. And I'll just quickly do my acknowledgements. I know I'm a bit over time, sorry about that, especially with Christmas awaiting. But really important to acknowledge everyone in my group who's played a key role in all of the data that are presented at various stages, especially Manisha, who's developed the explant culturing system here for us. And also everyone in SCRE, Richard and Gillian for the PDX platform, and Toby and Val and Sarah, who we've just got some funding to do a WNT-based project as well here, which we're very excited about starting next year. And Matt and Boris, who've been instrumental in providing some prostate cancer models as well. And all the team at Peter Mac, which is where the whole story for PIK3CA originated. Special thanks to John, who's been really instrumental in helping ensure that all of my basic research is translatable into the clinic, and for giving us key directions on what to focus on. And Wales Cancer Bank, Johan at ICR for feedback, and Owen and Peter at the CRUK Pearson, they're collaborators on my CRUK grant, and they provide an amazing support for a lot of the molecular characterization. Teresa Marafiotti is doing the multiplex immunostaining for us, AstraZeneca. We've got lots of support there for preclinical studies for DNA damage repair, but also the Pi3 Kinase inhibitors, and also Ronald in Germany for the tissue microarrays. And yeah, and just really thankful for funding. I'm so sorry for going over time. I'm happy to take any questions.
Helen, thank you so much for that talk. Part of the reason for setting up these webinars was so that we across Cardiff University could get to know what each other's doing, not just in terms of the science, but also in terms of experimental approaches and infrastructure and so on. And so your talk and Kevin's actually have been fantastic in that regard, really served to sort of inform the community of some really nice mouse models down there, and the explants and so on. Brilliant, thanks a lot. That's great. So questions can come through on chat or on the questions and answers. And I have one which relates to, you mentioned, I think, right at the beginning of your talk, this question came through quite early, about prostate cancer incidence increasing. And the question is that related to, the fact that we are an aging population, or is it independent of that?
Well, I wouldn't say that that's not involved, because we are an aging population. I suspect it's more to do with the fact that we've got more uniform systems in place for detection, like digital rectal examinations and things like that are just more commonplace. And the charities have done an awful lot of work in trying to raise awareness for prostate cancer. So getting an older male to go and see their GP and get their prostates checked if they've got symptoms, that sort of thing is, that's improved dramatically now. So,
do your models, Helen, let you look at things like acquired resistance? So when you use a certain inhibitor, do the models allow for you then to look at what the effect of that is on, cancer progression and avoiding that drug?
Yeah, absolutely. So we can do that in vivo and ex vivo, but a lot of the work that we've done so far looking at acquired resistance has mainly been in vivo, so that we can actually see the progression as things change. So we'll take, for example, a tumour model and castrate that, and we take multiple time points. So you'll get the initial response 3 days in, 10 days later, 2 weeks later, and long term. So you get the range of how those models are responding to treatments. And it's really important because it gives you a really useful insight into what's actually happening during those acquired resistant phases. The transition to CRPC is so crucial, trying to understand the molecular mechanisms that in play is really what would be, I guess, paramount to develop new actionable targets for the treatment of CRPC, or avoid it even, or develop biomarkers to identify those that are not going to respond.
And that sets us up nicely for telling you that John Chester said, "Thank you very much for relating your lab research so nicely to the clinical situation, Helen. Bravo."
Try my best. I mean, I think that's what comes into play being at Cardiff, because you're so close to the hospital. And it was the same at Peter Mac. I mean, it is actually a cancer hospital, so you're in constant connection with clinicians. And I think that's, for my research and for lots of other people around ESCRI as well, it's essential to know that you're on the right track in terms of what the clinic needs. So yeah, which is why I was very, very grateful to John, and also to Howard, who I didn't get a chance to mention because I was rushing through my acknowledgements, but Howard's been really useful as well in underpinning what we do. And also to Rob Jones, who's been fabulous, because he's just set up the FACTION trial for breast cancer, looking at AKT inhibitors for breast cancer. And obviously I'm very interested in that in prostate, and we're actually, Cardiff's going to be a new site for the Capitello trial, which is AKT inhibitor for metastatic hormone sensitive prostate cancer, which I think will be really valuable. And we're talking with AstraZeneca about accessing tissue samples so that we can test some of our novel ideas on samples from the clinic as well.
And John Stafford has noted that the lab clinic engagement is particularly important now as the clinic is changing so rapidly. So with that, I guess the major challenge is patient selection and finding the right combinations of approaches, right?
Absolutely. Yeah, I think that's always going to be the challenge, because the treatment landscape evolves all the time. So, and again, for the models, that's the problem too, because we've got models that are outdated because the tumours don't really reflect what we see in the clinic today anymore. So developing new models continuously is probably going to be one of those like never-ending challenges, so that you're trying to capture models as the treatment landscape changes too. It's an ongoing process, but yeah, you're right. Getting the patient stratification side of things assigned, it will be key, I think, to finding the right therapeutics and improving precision medicine approaches.
Brilliant. I think that's a fantastic way to end the academic year, Helen. Thank you ever so much for highlighting, the importance of that lab clinic axis, and for showing how well it's working here in Cardiff. Absolutely. So before we end the webinar, can I ask you, Helen, to pop up the last slide, which is just to give everyone some dates for your diaries for next year. So these webinars will be held same time, Friday afternoons, 3 o'clock. More information will follow, but just as a, as something to put down in your diaries right now, Kate Brain will speak in January about the work that she's been doing, looking at how COVID has impacted on cancer care in Wales and beyond. And then in February, Dr. David Hughes, who's the director of the Welsh Cancer Intelligence and Surveillance Unit, will talk to us about their activities, which links, perhaps nicely with the stuff that Kevin was talking about. So on that note, happy Christmas, everybody. And I guess we'll have a few more of these seminars held as webinars, but, there is hope that at some point next year, these will actually be held in a face-to-face setting. And at this point, we would be going off now for a glass of wine. In the meantime, enjoy the holidays, and we shall see you next year. Thanks ever so much, Helen and Kevin. And bye, everyone.
Thanks, Owen.
Bye. Thanks both. That's great. Really good.
Bye. Thanks, Owen. Have a nice Christmas.
Thank you. Happy unpacking. Bye.
Thank you.